Today's topic is preimplantation genetic diagnosis and preimplantation genetic screening, and what are the difference between these two. I am Dr. A. Kazi. I am retired professor, head of the Department of Obstetric Gynec, WIMS Bellary. I also worked as DNB professor for 8 years, and I also received International Society of Scientific Network Research Award. So the topic covers preimplantation genetic screening and preimplantation genetic diagnosis. What are the difference? And also non-invasive prenatal test. Preimplantation genetic diagnosis is done where the parents are carriers of genetic abnormalities, autosomal diseases, X-linked. They are parents, so they want to detect whether the fertilized ovum is having the carriers or not, and select the ovum which is normal. So preimplantation genetic diagnosis, here it is done where there are autosomal parents are carriers of autosomal diseases, recessive or dominant, and X-linked diseases. Autosomal diseases like thalassemia, sickle cell anemia, and others, whereas X-linked involves hemophilia, where females are the carriers and males are the sufferers. So hence in this also they will select the X ovum, fertilized ovum, so that they are only carriers and not sufferers. So here there are various procedures done on the fertilized ovum. So one is blastocyst biopsy. So 8 cell or 12 cell stage they will do blastocyst biopsy, and troph ectoderm biopsy. These are the two times when these are done. So here they will select the fertilized ovum which is normal. For example, if the parents are having a hemophilia, so they will select the X ovum which is having X chromosomes, XX, so that they are carriers and not sufferers, and autosomal recessive diseases, and they will select the ovum, fertilized ovum, which is not having carrier of these diseases. Now there are various techniques for knowing this, and whereas prenatal genetic screening, preimplantation genetic screening involves, it is also called as aneuploidy screening. Here parents are genetically normal, but the problem is they are having repeated abortions, implantation failures, or abnormal babies. So they will detect what are the any abnormal chromosomes, and they will try to implant the normal fertilized ovum. So this is the preimplantation genetic screening, where parents are genetically normal, but there are implantation failures in IVF or normal, and implantation failures, repeated abortions, or abnormal babies, and this is also called as aneuploidy screening. Now what are the various techniques involved in diagnosing this? One is fluorescent in situ hybridization. Fluorescent in situ hybridization, which will 23 chromosomes which are, they can detect abnormalities. This takes 8 to 10 hours. Whereas next is comparative genomic hybridization. This will take at least 18 hours. So this is more specific. So this involves cryopreservation of the fertilized ovum. Then other tests are polymerase chain PCR reactions, and single nucleotide polymerization. So this will take 3 to 4 days, and which requires the fertilized ovum to be cryopreserved. But these are more specific. So these are the indications and techniques for doing preimplantation genetic diagnosis and screening. So for example, hemophilia is a X-linked disease, so females are the carriers, so they will select the fertilized ovum which is having XX chromosome, female, and they will implant it. Now what is non-invasive prenatal test? Non-invasive prenatal test, here depends upon cell-free DNA in the maternal blood. So placental, so there is there occurs degenerations few parts, and DNA is liberated. So that can be detected, cell-free DNA it's called as. So this will be useful, especially in and even in pre-implantation genetic diagnosis. Also HLA typing. HLA typing can be done, so that organ donors, if in the family it can be like thalassemia or other thing, can be HLA typing can be done. Non-invasive prenatal test, RH typing of the fetus can be done. So these are the indications for the procedure.